So, as always, let's try and get started here with our clicker questions. And maybe we can actually see it now. Yes, we have our countdown. Wonderful. The interaction between a virion and its receptor, quote unquote, determines tropism, in which subcellular compartment the virus replicates, MOI, number of virions for PFUs, or the eclipse phase. Okay, let's stop. What do we think? Yay! Everybody's right. Hallelujah. Pardon? No, I'm not quite sure why that. No, it seems to be counting up for these, which is strange. I'm not quite sure why this is still doing this. It started again, so that shouldn't be the case. Stop this one. Okay, question 2. It doesn't like this either. Okay, well, the answer is still A, tropism, and 91% of you had that right, so the rest of these we can kind of ignore for the time being. Let's see, I'm still trying to get used to this technology. It's trying to change too many different computers on me here. Pardon? It's trying to do question 2. That's because I've been clicking this thing, obviously, too much. So let's try question 2 then. The real question 2, as opposed to the partial question 2, which, assuming this computer actually talks to me, would help. Of course, now it's decided to give up completely. Let's see. Yay. Okay, so we'll start this one now. Good. Okay, we started. Everyone happy? Yes? In virions whose membrane fuses with the endosomal membrane, the trigger for fusion seems to be receptor binding, case change, conformational change of fusion protein before fusion peptide insertion, conformational change of fusion protein after fusion peptide insertion, and endocytosis. I say or endocytosis. A minute and 30 seconds, so 90 seconds. And feel free to talk to your neighbors. It's supposed to be interactive. Okay, so we had 54 answers, and no, these don't count yet, but I am planning on having them count next week. Virions whose membrane fuses with the endosomal membrane. The trigger for this fusion seems to be, most people think B, but we've got quite a few other ideas in here as well. There are 2 keys to this question. The first one is fusing with the endosomal membrane, not fusing with the plasma membrane. Fusing with the plasma membrane, receptor binding would be correct. The trigger for this, so what sets it off, is really the pH change. So now if we do, let's see, select, one more. Yes, that's the correct one. And then the conformational change that happens, and this is before fusion and after fusion takes place, this conformational change is due to the pH change, but it's not what's the triggering of the fusion event. So that triggering is really due to the pH change. And then the afterwards, that happens once you've already had that insertion event that takes place. And of course, endocytosis is happening right at the beginning of this whole process. So now we'll switch computers very quickly and move on. Today we've actually got sort of two lectures in one, really talking about bacteriophage to a very small extent, and mostly this has to do with entry. This is still the entry aspect of things. Talk a little bit about antiviral drugs and some defense mechanisms, which include P53 and PKR as far as eukaryotic cells are concerned, and the CRISPR system as far as bacteria are concerned. Anybody go to the biology seminar yesterday? Absolutely awesome. Really cool stuff about what PKR might be doing, as well as being involved in viral defense. Might have to do with metabolic disease. Really fascinating stuff. But first, virus entry in bacteria. The key with virus entry in bacteria is that viruses are actually really hard, bacteria I should say, are really hard to get into. It's really a challenge to get into any particular bacterium. They're gram-negative, they've got one membrane, but they've got a cell wall that you need to get across. If there's a gram-positive, they've got one, they've got a cell wall. Gram-negative, they've got two membranes that actually have to get across. So how do you get through this? And most bacteriophage end up doing this by literally injecting their genome through the membrane, which is really pretty amazing, and we'll see the video in just a second here. Once inside, they're good to go, because bacteria, at least most bacteria, don't have any kind of internal components. So it's not like the case in most of your eukaryotes, where you have to get into the nucleus in order to replicate. Once you're in the cytoplasm of a bacterium, everything's there. You're good to go. So now let's take a quick look. I apologize to those of you who have seen this video too many times before, but I just love it so much that I have to keep showing it. Plus, we've got a couple of visitors today, at least one, and it's a very cool video. So, and we had a question last time about how real this is. This is completely based on experimental data, the whole process here. The soundtrack that goes with it is not, which is why I've turned it off. So you have receptor interactions that happen with the tail fibers, the long tail fibers of T4, conformational change that happens. Once that conformational change happens, then you literally inject your genome through the first membrane. It releases proteins that degrade the internal membrane, and then it's actually been degraded in that process. And yeah, this is our friend Bacteriophage T4. One of the people, in fact, made that movie told me there's a big problem with it. Anybody notice it? No, the contraction's going counterclockwise rather than clockwise. So again, this gives you the kind of idea about how much they've really gone through to try and get a good idea what's going on here. Yeah.
So the proteins that it injected to degrade, you said the inner membrane?
Mm-hmm. Yeah.
How do they get through the cell wall? Do they bore through?
They're literally bored through with that injection process, yes.
And do they only degrade like part of the inner membrane there, or?
Yeah, it's just a very localized degradation, and then I won't get into all the details. If you're interested, we can talk about it offline. It's pretty well known for Bacteriophage T4, exactly how that happens. So that's how the bacteria get in. We had all the eukaryotic viruses, say bacterial viruses get in. We had all the eukaryotic viruses just a second ago. Talk a little bit about viral therapies, and this is why people love Tamiflu, etc. How do you deal with virus infections? There are lots of different ways of doing that. We're particularly going to talk about those viral therapies that have to do with that really early process, virus binding, entry, and uncoating. And there are a number of different drugs that work in this process. Blocking of binding, okay, you've got drugs and people are trying to get soluble receptors. So if you just flood the system with what the virus is going to bind to, then the virus will bind all these things. It's not a cell, and so it won't actually be able to interact with the real receptors on the real cell. Great idea in theory, hasn't worked yet. Basically, vaccines work by blocking binding because the antibodies which are being produced due to vaccination will just bind up the surface of your virus. And very often, it's the receptor binding proteins that the antibodies are reacting with, which makes sense. Those are the things that are sticking out of the virus particle. So, standard, and of course, vaccine, didn't really talk about it too much. We'll talk about it more later, is by far and away the most cost-effective way of dealing with virus infections. So get your vaccinations. If you've got issues with vaccinating your children, come and talk to me. I will try and convince you otherwise. The other thing, and this is much more of a drug-related way of dealing with viruses, is literally trying to inhibit the uncoating of the virus itself. And of course, these are happening in different ways, so there are different drugs that are dealing with this in different potential ways. But one of the best known are the so-called M2 channel inhibitors. This works well, or I should say it used to work well against influenza. Now almost all influenza have been evolved away from being sensitive to these M2 channel inhibitors. But we'll take a look at that. The M2 channels, we'll talk about much more when we talk about influenza later on, is important for sensing that pH change, what's happening inside the endosome. So we'll see what that is. Rhinovirus uncoating. Turns out that rhinoviruses are very similar to the picoRNA viruses, and particularly polio. So we'll look at some of how these are actually blocking that particular interaction. Take a look at the first one. This is amantadine interacting with the M2 channel, an influenza virus. The M2 channel is in the virus membrane. And so it actually normally will let hydrogen ions into the interior of the virus. Through this channel, there's your molecule right there, which is actually blocking it. So that's one way that you can block uncoating, because it just has to do with your acidification process. Also have binding to surface proteins of various different viruses. This is poliovirus, classic picoRNA virus. This is the drug right here binding to the VP1 protein. VP1 is one of the three proteins that making up each of those triangular faces of the icosahedron. That's where you have the binding to the poliovirus receptor. That's where you have your threefold axis of symmetry. And you have to have a conformational change to release the genome from inside these particles. And what these guys mostly seem to do, these drugs I should say, is block that conformational change. Once the drug is bound, you can't have the conformational change. You can't actually move in. So that's specific to the individual viruses. There are some drugs, bisindolylmaleimide. No, I don't expect you to remember that. Don't bother. But ways to deal with virus infection might be a good thing to remember. Just blocks endosome acidification. And so if you block endosome acidification, you're not going to trigger that pH-dependent conformational change that you have in the fusion peptides, so you're not going to have that change. Now, of course, if you block endosome acidification, you'll have other kinds of problems, because you do need the endosomal processes. Again, all the viruses are using these normal cellular processes for getting inside the cell. So that's an issue. Tamiflu, all of our friend here, is a neuraminidase inhibitor. Neuraminidase is that other protein that sticks out of influenza viruses on the outside. And we already talked a little bit about neuraminidase. What neuraminidase does is it cleaves the sialic acid off of whatever that sialic acid is bind to. And that means that the hemagglutinin, which we already talked about last time, can now be released, and so the whole virus can go inside the rest of the cell. So if you have too much hemagglutinin and not enough degradation of that binding, the virus is clump. And that's the whole basis for the hemagglutination assay, which we talked about right at the very beginning. Neuraminidase cleaves that last sialic acid off and allows the virus to get inside the cell. That's why if you have very large amounts of virus, your low dilutions in your hemagglutination assay, why you don't see agglutination, because the neuraminidase has broken things down. We know what the structure of this looks like as well. This is also Tamiflu is just another name for exactly the same thing, bound to the neuraminidase enzyme. This neuraminidase enzyme, the active site is right here in the middle. This is where it would be binding to the sialic acid and chewing it off. Turns out that this, all of you I'm sure know in your space-filling model for sialic acid, right? Looks a lot like this, only if it's, of course, not cleavable in that particular case. So that's it for the strategies for how to deal with some of these viruses. Really quickly, how do viruses get out? Either blast open the cell completely. A lot of bacterial viruses, particularly Bacteriophage T4, do this. In general, this is true for the non-enveloped viruses. It's really easy to do. How do you do it? You express something in your late genes that causes the cell to blast apart. So that works really quite well. The big problem with this nice, simple strategy is if you start expressing these lysis proteins too soon, you blast open your host before you have a chance to make any more virus. So that, of course, is going to be really, really critical, and we'll talk about that for the small RNA bacteriophage in a couple of minutes now. On the other hand, you can do budding, and this is true for almost all of the enveloped viruses. Again, there's always exceptions to these things. But an enveloped virus, as you remember, it's got the membrane around the outside, and very often it's picking up that membrane from the plasma membrane of the host. And budding, the whole idea is you get membrane fusion when you bud off. Well, you fuse your membrane back together of the host, then that membrane's fine, and the host can keep producing more viruses, etc. So the timing is nowhere near as essential for budding as it is for lysis. So you can sort of, bud as much or as little as you like. The timing is not really critical there. To get this appropriate budding process, however, you have to get all of the virus envelope proteins to the right place in the cell membrane. So you have to get them all together and then get either your virus genome or your virus nucleocapsid to the right place. And so that's certainly much more of an issue, but at least it's not trying to get a three-dimensional assembly. Should have brought my soccer ball today. But it's just getting things assembled in a more or less a plane, at least as far as the virus is concerned, its size, the geometry of the plasma membrane. It's going to look pretty flat. So these are pretty much two-dimensional assemblies that are taking place. How do cells normally deal with this process? Well, usually in eukaryotic cells, it's just slowing stuff down. Slow everything down, allow, and in most multicellular eukaryotes, you have an adaptive immune system, which will then come in and take care of the cells that are infected with virus, or maybe even take care of those viruses itself. So the main thing that cells are doing in order to deal with viruses is just slow down. Slow down, wait, or even in some cases undergo apoptosis. So what are these cellular defenses? Apoptosis, clearly an issue, but you have to be careful about apoptosis, because what happens with apoptosis? Right, you lyse a cell. Well, it's a naked virus. It's exactly what it wants. And in fact, some viruses do stimulate apoptosis. So again, the timing here is really important. However, if it's an enveloped virus and you stimulate apoptosis, what's going to happen to that virus? It's not going to be replicated terribly well. So yeah, it depends on what kind of virus you're talking about. There are also mechanisms, and again, this is particularly true if you're thinking about the naked viruses, those that don't have the envelopes, not reproducing by budding, is just shut down protein synthesis. You think about what happens when the virus infects a cell. To totally over-anthropomorphize again, all the virus cares about is making more virus. And a lot of that is protein. We talked about the capsid proteins. We'll talk a lot more about capsid proteins later on. 90% of the cellular protein synthesis is going towards making more virus. So if you shut down cellular protein synthesis, you'll also shut down virus synthesis. You also, of course, shut down your own protein synthesis, which can be an issue as well. Last thing in terms of delay tactics, and if anything, this is sort of the adaptive immune system. Somebody was asking me about this a couple of lectures ago. In prokaryotes, the so-called clustered regularly interspaced palindromic repeats, god-awful name, but unfortunately that's what we've got, was originally discovered because there was this bizarre sequence in the E. coli genome. And that was it. Just a bizarre sequence in the E. coli genome. No one had a clue what it was. It was guessed in 2005 what it might be doing, and strangely enough, I was actually at the meeting and presenting right after the guy who talked about this. So just guess how many people listened to my talk. But so they thought that actually this might be functioning a little bit like RNAi in eukaryotic systems. And a couple of years later, it was actually really nicely demonstrated that it works at least in a few organisms. It's not been completely demonstrated in all organisms, but this has become a really hot research field in terms of looking at what's going on in terms of a lot of these bacteria. So let's talk about these individual mechanisms right here. Hopefully this is review from cell biology. And if some of you haven't had cell biology or taking it right now, okay, great review for cell biology. Only we're talking about it's a very small part of it here. The real focus here is P53. So P53 really has two different roles. One of its roles is just shutting down the cell cycle, stopping things from going through replication, etc., because there's some kind of problem. Usually DNA damage, cell cycle abnormalities, and we'll see also virus infection can lead to this. So if it's not too bad, and there's ways that P53 can sense this, then it just shuts down the cell cycle. You have enough time to do all your repair, maybe get rid of these viruses as the case may be, and then restart the cell cycle. On the other hand, if things are really bad, again, usually the case when you have a virus infection, particularly with one of these enveloped viruses, undergo apoptosis. So just kill off that one particular infected cell. If you're a multicellular organism, this is not a big deal, and you're good to go. This is everything you need to know for cell biology. So remember it for the midterm. Everything you need to know about for this class is right here. Red circle, brown circle. So the red circle, of course, is here P53, and P53 leading to apoptosis. And we'll see for a lot of the individual viruses, particularly some of the polyoma and papilloma viruses, that they interact very closely with P53 in terms of regulating this apoptosis event. So everything else here is cell biology. This is what we care about in terms of virology. The other defense system, and this is pretty specific to vertebrates, is a so-called interferon response. And anybody heard about interferon before? Tom, do you know what it does?
For hepatitis.
Yeah. So interferon was in fact originally discovered because it was interfering with virus replication. So that's where the whole interferon comes from in the first place. And again, most of this is cell biology. There are a couple of important things as far as the viruses are concerned. And of course, where are they? They're in the nice little red box, so you can find them easily. P53, which we already talked about, and a new player in here called PKR. So PKR is protein kinase stimulated by double-stranded RNA. What do protein kinases do? Phosphorylate things. So that's what we're talking about. We're talking about this. Double-stranded RNA happens almost always. In fact, it does always happen when you have a single-stranded virus that infects, a single-stranded RNA virus, because you have to copy that opposite strand. You always have to make a negative copy of your RNA. So you're always going to have double-stranded RNA in an RNA virus infection. DNA virus infections are a bit of a different story, and we'll talk about those when we get to those a little bit later on. So the idea here is you have a virus infection over here on the left-hand side. That then will give you double-stranded RNA, which leads through all of these different kinds of things and eventually turns on interferon. Now, the great thing about the interferon is it then gets exported from the cell, it's secreted, and then can go and tell other cells in the vicinity, hey guys, there's a virus around. Turn up your defense mechanisms. And so that's all that this is about. So interferon is produced here due to the virus infection, and then leads to expression of p53 and PKR to block any future virus replication. So it's sort of a defense mechanism saying, okay, we got this virus infection over here, quick, turn on interferon. That will tell our neighbors that something's going on. Yeah?
Have there been many studies on how effective these processes are? Maybe not apoptosis, but the interferon, is it pretty successful for, I guess, cell lineage or maybe for a cell?
So yeah, the basic question is, I think volume 3 of the textbook that we're not using. So yes, a whole bunch is known about it. There's all kinds of ways that the viruses are trying to deal with this system because they've been evolving at the same time as the defense systems have been evolving. So it's really this whole trade-off which is going on there. And yeah, if you're interested about it, take a look at it. In fact, there's this VNS1, which I'm ignoring over here. It's a viral protein that's involved in dealing with this response. So we'll talk a little bit more about that later on when we get there. But what is this PKR actually doing? So it senses this RNA virus infection, and all that it does is phosphorylate EIF2. Okay, what's EIF2? For what? Eukaryotes in what mechanism? What process? Translation. So, translation initiation. And so, phosphorylation of EIF2 blocks translational initiation. And so when you turn on PKR, you stop making new proteins. So it's a really nice way of stopping any kind of viral replication. Of course, there are viruses that have ways of dealing with this. We'll talk about a number of them. But you're also shutting down the cellular translation in the process. So it's a pretty blunt instrument in terms of trying to deal with the whole process. So the last thing I want to talk about in terms of viral defenses are these CRISPRs, clustered regularly interspersed short palindromic repeats, which is why everybody says CRISPRs instead of the whole name. These are really fascinating. Again, they were originally found because of sequence gazing. And really, you could say that this is a mechanism that was found because of the advent of genome sequencing. And what people found are these little blue things over here on the right-hand side. These are those regularly interspersed short palindromic repeats. I should have 5 of them so I can go through this whole process. So each of these are repeated sequences. And you find a whole bunch of them, maybe like tens to hundreds of them. So this was just bizarre. And that's all that people noticed, that were bizarre. And it was much later that what people noticed was that in between these repeated sequences were these spacer sequences. And these spacer sequences are relatively short. They're on the order of 30 or 40 nucleotides. But they match virus sequences. At least some of them do. So, okay, we've got all these repeated sequences, and then things that are matching small pieces of virus sequences. And then, which is what happened in 2005, where people noticed that there were these genes that were just upstream of all of these spacers and repeats that looked like proteins that were involved in RNAi in eukaryotes. And so their thought was, okay, well, these guys get transcribed. You've got all of these short RNAs that could then match the RNA of an infecting virus, or in some cases could match the DNA of an infecting virus. Just like the case with RNAi, you've got a matching sequence. It will come down and bind to its complementary sequence, causing its degradation. So the prediction was, well, that's exactly what's going to happen here. You have a virus infection. It runs into some of these things that match will then cause degradation of those particular virus sequences. And that was a prediction in 2005, and lo and behold, in 2008, they actually showed that this is exactly what was going on. That you have these transcribed spacers, which are then giving you resistance to virus infection, again, at least in a couple of organisms. It hasn't been shown very well for a lot of organisms. But that is exactly what is going on in this particular case. So it's acquired immunity by bacteria, which nobody thought was possible. Now, the big question, and this is still a very open area of research, is how the heck do you get these spacers in the first place? So they've got to come from somewhere, presumably from a virus infection that was not actually successful. And you've picked up these little pieces here, which then provide you resistance to any future virus infection, which is going on. So in terms of, yeah?
How does the matching up cause the degradation, though?
Okay, so basically, how does matching up cause degradation? Let me back up here real quick. This process right here, basically what happens is you have these short sequences, which are now cut out of this particular repeated sequence here, because there are specific endonucleases that will cut and give you all these little pieces. If this little piece matches anything else, that's a target for it to get degraded. And it's just like the case with RNAi, is if you have your short RNA that's combined with various proteins, it could be dicer, it could be argonaut, then when you have this base pairing interaction that takes place, then that will get degraded. And it's pretty well known. It turns out that these are the proteins up here which are encoded by all these genes, which are doing that. But it's a really kind of cool story, and I've been involved with it kind of from the beginning too, which is really kind of fun. Turns out that the favorite organism, my favorite viruses, are really messy as far as trying to understand this way. So thought it'd be really interesting, but less so now. So in terms of what we've talked about for sort of the first half of the lecture, before we start to talk about these small RNA viruses and bacteria, recognition all about the tropism. Again, receptors interacting with the virus receptor binding proteins. How do you get into the cell? Usually it's a normal cell-mediated process, at least for all of those eukaryotic viruses, except if you're one of these guys who says, okay, now, this is not a cellular process. Injection of a genome of a virus is not a cellular process. It's a purely viral process. So you've got both, but again, certainly for the eukaryotic ones, it's almost always a cell-mediated process. For the bacteria, it's usually this breaking and entering. Again, this is from my graduate student when he gave this last year. Uncoating, this can either be happening literally through the membrane fusion that we looked at the videos about last time, or some perforation of that membrane, sometimes even uncoating of the virus particle itself. That can happen either earlier on in the process or literally right at the nucleus. And lots of different ways of getting into the nucleus. And then today we talk a little bit more about cell resistance. So there are questions about this part or last lecture before we move on and start to talk about, finally, specific viruses or specific virus families. Okay, so let's move on. Get this actually working here. 2 hands, good thing. So this is the review process again. Basically, it's what we talked about before. How do you get in? All cellular processes, and then literally getting inside the cell. So what are we talking about now? It's really about single-stranded RNA viruses, and the way the lectures are done, I also like the way the textbook is done. It's partly why I like this textbook, is sort of going from the simple systems to the more complex systems. And so we're starting with very simple viruses now of bacteria, and we'll move on through more and more complicated ones as we move through the rest of the course. Start out first with the bacteriophage. We haven't talked too much about that yet. ssRNA phages, some of the simplest of the bacteriophages. And the big sort of take-home message here is it's all about a problem between transcription and translation. And this is always going to be a problem if you're thinking about RNA viruses, because that RNA is serving as the genome, and it's serving as your messenger RNA. So the genome needs to get replicated, and the messenger RNA needs to get translated. How do you deal with these two things? And so that's a problem for all RNA viruses, and certainly for these single-stranded RNA phage as well. What's a phage? Phage from the Greek phagos, eat. Eats holes in bacterial lawns. So that's where they found it originally. Found also in the early 1900s, so it was very soon after tobacco mosaic virus and some of the plant and animal viruses had been found. So it was really quite early on that these were discovered, apparently simultaneously by a Briton and a Canadian. They argue about who discovered it first, but be that as it may, both of them usually get credit for it. And it was just about the same definition of viruses. Something you can get through one of these filters that blocks all bacteria that makes something sick. And in this particular case, it's the bacteria which are being infected. The bacteria are getting sick. And this was really the beginnings of phage therapy that I talked about first lecture, I think. Cool things you can do with viruses. So using the viruses to treat bacterial disease. And in fact, Félix Terrel was one of the very first people to do this. And he was working on cholera, actually, at the time, to try and understand and use viruses to treat cholera disease, which in fact does work reasonably well. There's this whole message, a real quick aside here. Drinking the water of the Ganges seems like a really strange thing to do. Well, it turns out that the water of the Ganges is loaded with bacteriophage that kill cholera bacteria. So it might actually be a good thing. There are all kinds of other things in the Ganges too. But that being said, there are a number, in fact there's a company called Gangagene, which is literally trying to find some of these specific viruses and the specific virus proteins. And they've been quite successful, in fact, in finding a lot of these things. So okay, that's a little bit of an aside. In Bangalore, if you're looking for a job in Bangalore, I know the CEO. But small RNA phage, this is something I'm going to try and do for each of the different viruses when we come up. Sort of, these are going to be the really basic things that we talk about, how they found it, what the structure looks like, a little bit about the translation and replication. This is a very specific case with the RNA structure which is involved, and again, this translation versus replication process. And something which is pretty specific for these small RNA phage are some neat biotechnology applications which people have been using. How do they find these small RNA viruses? Well, we already talked about the water from the Ganges, right? So, sewage treatment plants. If you're looking for viruses that infect E. coli, where's the best place to go? Well, you find a bunch of E. coli. So, sewage treatment plants, and in fact, almost all of these bacterial viruses that we're going to talk about come originally from sewage treatment or something along those lines. So, because they're infecting E. coli. And where do you find the E. coli? So, this particular one, 1960s, present at pretty high concentrations, anywhere from 100 to 10 million per milliliter. It's, in fact, non-pathogenic at all for humans. We'll kill off the E. coli, but it turns out we don't need much E. coli in our gut anyway. So it's actually a really great way of determining sewage contamination. You may have heard about the coliform bacteria. Go out to the coast, people saying, warning, don't go to the beach because the coliform are too high. Well, that's the coliform bacteria, the E. coli, but it turns out it's even easier to detect these guys, these viruses. So a lot of the time in terms of, people will close beaches because they found these viruses that are infecting the E. coli, but at least it's an indication that there's sewage and again, Ganges-like water which is there. So this is where they're found in the first place. It's still used in some cases for looking at sewage contamination. And the reason that they were found had to do with everybody at the time in the late 50s and early 60s. People were trying to find new viruses and new viruses that infected E. coli. And in particular, there was a group that was interested in F-factor dependent viruses that infected E. coli. So this was really pretty straightforward. You looked at viruses that infected an E. coli that had the F plasmid, one that didn't have the S plasmid, and then you just followed up on the ones that had these. They gave nice clear plaques, i.e. they killed off the host very nicely. We'll talk about clear plaques a little bit later on. There are two flavors of these small RNA bacteria, Q beta and F2. This one's a little bit bigger. One of the reasons that a lot of people got excited about these things is true for why a lot of people were working with viruses, particularly in the late 1950s and early 1960s, is because there's a way to get a lot of nucleic acid, and a lot of nucleic acid that was identical to all of the other nucleic acids, partly why viruses are really great tools. You get a lot of, in this particular case, RNA, and you can also get a lot of complementary DNA to that RNA later on, and we'll talk about that when we talk about the biotechnology at this point, probably next week. What do the genomes look like? The book makes a big deal about, wow, we can just do all this stuff with 2 proteins. We'll talk more about stuff that deals with, can replicate a virus with just half this amount of protein. So, but be that as it may, these are still very, very simple virus genomes. And all you need are these 4 proteins, and it turns out you can actually pretty much get away with 2 of them. They're from 5' to 3', maturation protein, coat protein, followed by the replicase, F2, those are MS2, they're extremely similar to each other, is this genome arrangement, has a lysis protein, which is encoded here in a different open reading frame. That's being shown down here at the bottom. Different open reading frame that this is being encoded by, whereas the Q beta has a different protein, which is not made in a different open reading frame, but it's actually made by read-through of a stop codon, about 6% of the time. And we'll see, as we move on through the rest of the course, there are lots of examples of read-through. There are lots of examples of viruses using multiple open reading frames in order to encode what they need. But here, the important things are really coat protein, replicase, what do you make your virus structure out of, how do you make more of your genome, and how do you get outside the cell. The maturation protein, as we'll see, is probably sort of serving a bit like a virus receptor. It's actually less well understood what the maturation proteins are actually doing. What does the structure look like? T equals 3. Should be hopefully really obvious what this looks like from this structure up here. How do we get T equals 3? Starting here, 5-fold axis of symmetry. Here's a 5-fold axis of symmetry. And now we'll turn our pen on. We hop from here to here. Is H of 1. We change directions. K of 1. Yes, happy? No, unhappy? Do it again, please. Okay, we'll change color now. Hopefully this is still being recorded with any luck. If not, I will try and add it to the other one. Okay, so we're going to go from a 5-fold axis right here. So this is 1, 2, 3, 4, 5. Sorry, 1, 2, 3, 4, 5. If I could count, that would help. Down to a 5-fold axis over here. 1, 2, 3, 4, 5. Again, these are our 5-fold axis of symmetry. Here's that 6-fold axis in between. So hop 1, change direction, hop 1. So H of 1, K of 1. Scribble so there's an H and a K. Drawing with mice is really hard. So T equals 3, icosahedral particle. What does that mean in terms of the number of copies of your coat protein? Just one coat protein. T equals 3. T times what? Gives you 180 copies. So it's 180 copies. 1 copy of the maturation protein as far as we can tell. And that means it's actually probably not exactly 180 copies because it's not going to fit in here properly. This is, in fact, a particle which was made in the absence of the maturation protein and also in the absence of the single-stranded RNA which was there. The coat protein by itself can actually assemble into these particles. And so when it says Bacteriophage MS2 up there, it's actually just the coat of MS2, which is being used there. How does MS2 interact with the cell? Well, it was originally isolated as being an F-specific bacteriophage. So something to do with the F protein. One of the things that the F protein does is encode for F pili. F is the fertility plasmid in E. coli. That's how E. coli undergo the nasty, as it were. So here, F. pili, which would then interact with another cell, usually will be an F minus, and then genetic material gets transferred normally through these pili. You now have an E. coli that's being infected by one of these single-stranded RNA-phage. These pili are completely coated, and you can see it better down here, with all of these icosahedral virus particles. So I should say virions that are associated with it. The maturation protein seems to be the really critical protein in terms of getting interaction with the pili. If you make these particles in the absence of maturation protein, they don't associate with pili. And then once you have this association, the maturation protein is cleaved. Should sound really familiar. Cleavage, proteolysis happens all the time in these virus receptor proteins. And then somehow you get inside the cell. These are really nice small viruses. They don't have this wonderful injection mechanism like you have with Bacteriophage T4. So how the RNA actually gets inside the cell is not entirely clear, but we know that when conjugation takes place between an F plus and an F minus E. coli, those pili actually retract and pull the two cells together. And so it's distinctly possible it's in that retraction process that you bring the RNA inside the cell. So again, it's not entirely clear. That's why we got the question mark here at the end. Oops. So now we've got our RNA inside the cell. We immediately have our classic problem with RNA viruses. You've got a ribosome that's going to want to translate and give you the proteins that you need. It's just the RNA that's come inside the cell. At the same time, you need to be making a bunch of new virus genomes. So these guys, boom, run into each other. So how do we deal with that? That's one problem. The other one is, this is true again for all viruses, you need a whole bunch more of your coat protein than you do of maturation protein, or for that matter, replicases. We just got one RNA. How do you deal with that? How do you figure out that you're going to get a whole bunch more protein and a lot less of these guys? Well, the main way that this happens is it's a difference between translation or replication that takes place. And it's all about the secondary structure of your RNA. So whenever we talk RNA, it's almost always going to be about secondary structures, which are forming in the RNA, which are going to be regulatory. In the case of these small RNA viruses, basically MS2, F2, Q beta, more or less identical to each other. We have the start codon and the Dargarno sequence, which is where the ribosome is actually going to bind to that RNA. For the coat protein, which is not in this massive secondary structure. Turns out it's pretty available. The ribosome can jump on here and start to translate immediately. On the other hand, the lysis protein start codon, the replicase protein start codon, as we'll see later, the maturation protein start codon, are really hidden. They're in these secondary structures. So here, the so-called operator hairpin. This is right when the operon model was coming out for gene regulation, so they use the same terminology for that here. So the operator hairpin is what's regulating the production of the replicase, and it turns out also the lysis protein as well. So you have binding to this start codon for the coat protein gene. It then leads to translation of that protein. All of these other guys are completely shut down. And one of the main things, I've just circled it here in red, if you make mutations in this particular sequence here, where you lose the secondary structure, then you start to get translation of replicase, and a little bit of the lysis protein as well far too early in the process. Replicase is not such a big deal. Remember, lysis protein, this was going to blast open the cell at the end. If you start making that too soon, it's a real problem. Just like we talked about virus release just a second ago. That something has to be absolutely critically timed. Because if you break open the cell too early, you're not going to have any of your virus genome. So what are we looking at here? Here's our elongating ribosome, started from the coat protein start codon, chunking along your RNA, making coat protein. As the ribosome translates, that ribosome will actually cause these secondary structures to be released. So the more of this coat protein you get, eventually you're going to be able to release this replicase start codon. So lots of coat protein start to make replicase. Makes sense. You could, in theory anyway, use just that one RNA that gets inside the cell to make all of your coat protein. But in general, you're going to want to have some extra genomes to use for that as well. So here we are going through. On the other hand, once you've actually got enough of your coat protein, then you don't want to continue to make it. And so what happens here is the coat protein will also bind to the secondary structure here in your genome, actually shut down coat protein production and lead to the production of the lysis protein. You remember, you want to have the lysis protein, just happens late. Once you've got enough coat protein, that can assemble into your particles, and then you can actually get formation of all these virus particles. Again, they're inside the cell. So if you have virus particles inside the cell, what part of the growth phase is that? But not outside? That's between the eclipse and the latent phase. That's when you have virus particles on the inside, but on the outside yet. How do you get them out? You make the lysis protein. The way that's happening is that you now have coat protein that's bound over here on this side, which is blocking the ribosome from moving into making the replicase, etc. But also, what it does is this ribosome doesn't always dissociate immediately once it gets to a stop codon. Everyone, I'm sure remembers from that other course that I taught in the fall. You have release factors that associate with the ribosome. Basically, you trick the ribosome until making a peptide bond to water. The whole thing falls apart, and you start again. This takes time. And it turns out that the ribosome sort of sits here for a little while, and it does actually bounce back and forth a little bit before it completely dissociates. And if you have coat protein that's bound right here, blocking this movement over to the replicase and making the replicase protein, because you've got enough coat protein, it's time to break open the cell and get out. Now, what this ribosome does is it backtracks to the beginning of the lysis protein. About 5% of the time. So it's relatively infrequent, but that's enough. And so that will then lead to production of the lysis protein. You produce the lysis protein that will lead to cell lysis, and we'll talk about that in just a second. So now we've talked about how you make all three of the proteins that you need mostly for the cell. We'll get to maturation right at the end. So coat protein, open start codon, can make a whole bunch of it. The replicase protein, you only start to make it after you've translated some of the coat protein. After you've translated enough of the coat protein, now you turn on the lysis protein, because it's getting that ribosome to move a little bit. On the other hand, how do you replicate? Well, there are a couple of things that you need to think about in terms of replication. The first one is that positive and negative strands in these viruses don't seem to stably base pair with each other, which is kind of different from what the biology seminar was yesterday about double-stranded RNAs that form. And the reason for that is all the secondary structure that already forms in a single strand. And so it seems that the single-strand secondary structure is more stable than the two strands being able to form relative to each other. And so that's actually really important because it means that you're not going to be blocking any kind of translation that could be taking place once you've actually made some of these opposite strands. And so the interaction between the two is not a permanent one. It's a very transient one, so not very stable. The other thing is that once you start to replicate, you don't want to have your replicase banging into the ribosome. It's going to be a big problem. So what happens, apparently even before you get very much of the replicase protein, before it even starts to replicate, it binds to the start codon of the coat protein gene. And so it blocks, and particularly on that DNA, which is going to replicate, it blocks any ribosomes from getting in. So at that point, you've got replicase bound. It can actually do its thing in the absence of having a ribosome. How do you get this strand? Really interestingly, at least to me, I'm kind of biased, is that you not only block the binding of the ribosome to the coat protein gene, but you also use other translation proteins in order to get replication to take place. And so you're basically taking the translation proteins and using them for replication, so they can't be used for translation. So you're not just blocking the physical interaction, you're also taking some of the other proteins that are really important for translation, which you need for replication. So the host proteins that are actually needed for this are EFTU and EFTS, which are the elongation factors for bacterial transcription. Bacterial translation. EFTU is the GTPase when you make the right peptide bond for translocation. EFTS allows the recycling of EFTU. It's a guanosine nucleotide exchange factor. So these guys are involved in translational elongation. The S1 protein, which is part of the ribosome itself, is also required for minus strand genome replication. Again, just as a quick reminder, plus strand, that's what's coding for your protein. Minus strand is a complement to that. It binds to the coat protein start together with that replicase protein. And it turns out that that part of the protein is important for, that part of the ribosome is important for getting translation to take place there as well. But it's also involved in now getting your replication of that second strand. These are all of the standard regular proteins that are involved in cellular processes all the time. There is a so-called host factor. That host factor was found classically the way the geneticists always do things, breaking something and then trying to figure out why it broke. So they find mutants of E. coli that can't replicate these single-stranded RNA phage, and then they figure out what that protein is. Turns out to be a poly-A binding protein that binds to the 3' end of the virus genome. 3' end of the virus genome is CCA. You replicate the minus strand, and people said, okay, well, it's going to start with what? So what's going to base pair with A? RNA virus. U. So U, GG, etc. Strangely enough, it actually doesn't do that. It starts with guanine. And we'll also see that replicating ends of RNA genomes is just as hard as replicating ends of DNA genomes. Ends are problematic. Probably what's happening here is that this guanosine residue is coming from one of these GTPases. And so that's how you're actually getting started. That's probably why you have EFTU together with these host proteins all together. And then, once you get all the way out to the other end of the genome, then you'll add a residue. This is true for many DNA polymerases and also replicases as well. So that's how you make your negative strand. You need all these extra host proteins, make that negative strand or the minus strand. Now you need to make a bunch of plus strands. Well, plus strands are great because you're just using that minus strand as a template. The minus strand doesn't serve as a template for translation. So you don't have to worry about things banging into each other anymore. You can just make a whole bunch of copies of that negative strand template, and you just need the replicase to do that. Because you don't have this same problem. So you end up with a lot more of your positive strand than you have of the negative strand. It seems that there's some secondary structures that form in the negative strand as well at the end of the genome. Spend the last 5 minutes talking about the maturation protein. Said we already talked about 3. You need 4 proteins completely, and the maturation protein is involved in what? It's Friday morning. I need to get through the week. So it's interacting with the receptor. That's what's really interacting with the pilus protein itself. The maturation protein, you only need a very small amount of it. You only need it at the end of the whole process. It turns out that the maturation protein is only made from new genomes. That's what I mean by nascent genomes here. New stuff, stuff which is just being made. How do we know that and what's going on here? Well, it has to do again with, surprise, surprise, these are RNA viruses. What are we talking about in RNA viruses? We're talking about secondary structure. If you just look at the secondary structure of literally the very end of the genome, this is the 5' end of the genome, this is the very beginning of your genome, it makes this incredibly stable structure. And a very stable structure which includes the Dargarno sequence, and it's quite close to the start codon for the maturation protein. But the big issue here is this Dargarno sequence, which is what the ribosomes can end up binding to, well, the S1 protein of the ribosome, which is involved in that. So here, you can't get any ribosome binding. On the other hand, when you've just started to make this positive strand, again, just with the replicase, now this secondary structure over here hasn't formed because there's a different secondary structure which can form, which is pretty stable. In that secondary structure, you now have a free Dargarno sequence. And so the newly made positive strands can serve as templates for the ribosome, but at this point, you're not going to have ribosomes and replicases running into each other because the replicase is cruising off ahead here because it's using that negative strand. So it's not going to be an issue. Basically, the ribosome is actually following the replicase in this particular case. So now we have all 4 proteins. We just need to get outside of the cell. Capsid protein, find it as replicase hairpin. That seems to serve as a packaging signal, how to get that particular RNA inside all the coat proteins. You do want to just be packaging the RNA of your genome. You don't want to be packaging just any old RNA, which happens to be inside the cell. Most RNA viruses have these packaging signals. You can get assembly of the capsid protein in the absence of the RNA. You can also get just purified RNA, purified capsid protein, and get those nice T equals 3 icosahedral structures. That's what we saw. We looked at the structure a little bit earlier on. But of course, usually has the maturation protein associated with it. And the lysis protein, we've been making more and more of it because the ribosome's gotten blocked, then shifted back and is translating this lysis protein. You make enough of this lysis protein, it literally makes a hole in the cell, and then all of your virus falls out. And it turns out it's these lysis proteins that they're really concentrating on in gangagene.